Look at the sea star on that muscle. Not the whole animal, just the underside, where the arms fold down over the shell. It's been sitting like that for hours. The tube feet have it locked, hundreds of them, pulling in a direction the muscle was never designed to resist, and the shell has opened, barely. A crack you could miss if you weren't looking for it. Maybe the width of a sheet of paper, if that. That's all the sea star needed, because the thing coming out of the sea star's mouth right now sliding into that crack is its stomach, not a tongue, not a proboscis. The actual cardiac stomach, everted, turned inside out through the mouth on the underside of the disc and pushed into the gap between the shells. The muscle is being digested inside its own shell by an animal standing on top of it, and the sea star's stomach is currently outside the sea star. Now, you've probably heard that before. It's one of those facts that shows up in every tide pool talk. Sea stars push their stomachs out. Great. Weird animal. Moving on. But nobody ever asks the obvious follow-up question, and the follow-up question is the whole story. How does that work in water? Think about what digestion actually is for a second. Digestion is chemistry. You make enzymes, which are proteins, and proteins are expensive. You have to spend nitrogen on them, amino acids, energy, the whole budget. Then you put those enzymes somewhere near the food, and you wait, and the enzymes chop long molecules into short ones, and then you absorb the short ones. Every part of that assumes a container. Your stomach is a container. It holds the enzymes at a useful concentration. It holds the food where the enzymes are. It holds the products of the reaction until your intestine can pick them up. Take away the container and the chemistry still works, technically. It just doesn't work for you. So here's the problem. And it's worth doing the arithmetic on because the numbers are worse than people assume. Say you release a digestive protease into open seawater. It's a protein, maybe 30,000 Daltons. Nothing exotic. Left alone in still water, that molecule wanders by diffusion. And diffusion is slow. Really slow. In 1 minute, a molecule that size spreads out to roughly 0.2 of a millimeter from where it started. Give it a full hour, and it's managed about 1.5 millimeters. That sounds like good news. If the water is perfectly still, your enzymes stay basically where you put them. Water is never perfectly still. Take the gentlest current you can imagine on a rocky shore. 1 centimeter per second, that's about the speed of a slow drip running down a window. Barely a movement. In that same hour, that current has carried water 36 meters. Call it 120 feet. 1.5 millimeters of diffusion. 120 feet of drift. The current beats the molecule by something like 20,000 to 1. That's the whole problem right there. In the ocean, if you let go of a molecule, it is gone. It won't drift off slowly in every direction. The current picks it up and carries it, all at once, one way. And it gets worse when you look at concentration instead of distance, because concentration falls off in 3 dimensions. Picture the enzyme cloud as a little half bubble spreading from a wound. Let it grow from 1 millimeter across to 1 centimeter across, 10 times the radius. The volume goes up by a factor of 1,000, which means the concentration drops to 1,000th of what it was. Enzyme reactions run at a rate that depends on how much enzyme is present. Halve the concentration, roughly halve the rate. Drop it to 1,000th, and the reaction hasn't slowed down. It's stopped, in any sense that matters to a hungry animal. So the animal that digests outside its body is doing something that looks, on paper, insane. It's taking the most expensive molecules it makes and throwing them into the largest, most efficient dilution machine on the planet. And then there's the other half of the disaster, which almost nobody mentions. You're not alone out there. Whatever you dissolve, you've just made available to everything nearby. Amino acids, sugars, free fatty acids, all of it in solution, all of it drifting downstream in a plume, Bacteria live on exactly that. So do a lot of small animals. You spent a week's protein budget on enzymes, you liquefied a clam, and you fed the neighborhood. Digesting in open water is like cooking a meal in an unwalled kitchen in a wind tunnel, while a crowd stands downwind with their mouths open. So every single animal in this video, every one, is solving the same 3 problems. Write them down, because everything that follows is a variation on these 3. 1. Hold the enzymes in place at a concentration high enough for the chemistry to run. 2. Hold the meal in place so it doesn't wash away or get taken while you're working. 3. Get the product back, which is a separate engineering problem from the first 2, and it turns out to be the hardest one. That's it. That's the containment problem. Hold the tools, hold the work, recover the product. And what I find great about this, once you start seeing it, is that the ocean has solved that problem about 8 different ways. Not variations on one trick. Completely different architectures, from animals that aren't even remotely related to each other, arriving at the same set of requirements from completely different directions. Some of them cheat. Some of them build. One of them dissolves rock. One of them injects. And one of them, at the far end, has given up on digestion altogether and just absorbs food molecules straight through its skin like a sponge in a broth. We're going to work up that ladder in order, easiest solution to hardest. And the ranking isn't arbitrary. There's a real reason the order goes the way it does, because the first solutions on the list don't build anything at all. They steal a container that already exists. Start with the cheapest possible answer to the containment problem, and the cheapest possible answer is this. Don't build a vessel. Find one. Back to the sea star. That mussel shell is, structurally, a very good reaction vessel. It's rigid. It's watertight when closed, which is the entire point of a bivalve. It's got a hinge on one side and an adductor muscle holding it shut. And inside, there is a sealed chamber containing exactly one thing, which is the meal. The mussel built it. The mussel maintains it. The mussel spent its own calcium carbonate on it. And the sea star is about to use it as cookware. That's not a metaphor I'm stretching. Watch what actually happens. The sea star doesn't open the shell wide. It doesn't need to, and it can't anyway. What it does is get a grip and pull steadily, with a few hundred tube feet, each one a tiny hydraulic cylinder run off the water vascular system, and it just holds. For hours. The adductor muscle of a mussel or a clam is seriously strong, far stronger, pound for pound, than the pull a sea star can generate. In a straight contest of peak force, the mussel wins easily. The sea star turns it into a contest of endurance instead, and it brought a completely different kind of muscle to that. Bivalve adductors have what's called catch muscle, which can hold a shell shut for a long time at very low energy cost, but it isn't free. Nothing is free. Hold long enough and the muscle fatigues, and the seal slips, and when it slips, it doesn't fly open. It gapes, a fraction of a millimeter, sometimes a tenth of a millimeter, which is about the thickness of a sheet of printer paper. That's the moment everything is built around. Because a sea star's cardiac stomach is a thin, loose, folded sheet of tissue, and a thin, loose, folded sheet can go through a paper-thin gap. There's no rigid part. Nothing has to be forced. The tissue is pushed out by fluid pressure and body wall contraction. It finds the crack, and it slides in. And once it's in there, look at what the sea star has just acquired. It has enzymes, held inside a sealed calcium carbonate box. It has the meal, held inside the same box, unable to swim, unable to drift, unable to be stolen. It has the products of digestion collecting in a small volume, where its own absorptive tissue is already sitting. Hold the tools, hold the work, recover the product, all 3 solved at once by an object the prey paid for. I think that's about as elegant a piece of freeloading as exists in biology. The sea star has outsourced the hardest part of the engineering to the animal it's eating. And there's a detail here that I keep chewing on. Sea stars didn't evolve a way to break shells. Plenty of other animals did. Crabs crush them. Rays crunch them. Some fish just swallow them and grind. All of those are brute force answers, and they all have the same downside, which is that when you break the container, everything inside it is suddenly in open water. The sea star's answer preserves the container. It's slower. It takes hours instead of seconds. But at the end of it, nothing has leaked, nothing has been stolen, and nothing has been diluted. Which raises the obvious question about what happens when there's no shell to borrow. Because most food in the ocean isn't conveniently packaged in a rigid, sealed box. Most of it is spread flat across a surface, or suspended in water, or locked inside a mineral, or armored with glass. And every one of those forces a different answer. Let's take them one at a time. Stay on the sea star a minute longer, because there's machinery here that deserves a proper look. Pushing your stomach out of your mouth sounds like a thing that would just happen, mechanically. Squeeze the body, out it comes. And that's roughly true for the pushing part. Sea stars run on water. There's a whole hydraulic network in there, the water vascular system, feeding those hundreds of tube feet, and the body cavity itself is a fluid-filled space that can be pressurized by the muscles of the body wall. Raise the pressure, relax the right tissue, and the stomach turns inside out through the mouth like a sock coming off a foot. The interesting question is the other direction. How do you get it back? Because that averted stomach is now a loose, floppy, thin-walled bag of tissue wrapped around a meal on the outside of the animal, in the ocean. There's no rigid skeleton in it. There's no obvious handle. And whatever pulls it back in has to be reliable, because a sea star that can't retract its stomach is a dead sea star. The answer turned out to be chemical, and it took a long time to pin down. Work at Queen Mary University of London, published by Dean Semmons and colleagues in 2013, identified a molecule in the common starfish. Asterias rubens, that does exactly this job. They called it NGFF-yumide, after the sequence of amino acids in it. Applied to isolated stomach tissue in the lab, it makes that tissue contract. Injected into a living animal with its stomach out, it pulls the stomach back in. And there was an older half of the story already sitting there. A family of starfish neuropeptides called the salm-famides. The first two of which were pulled out of asterias decades earlier, do the opposite. They relax the cardiac stomach, put them in, and the stomach goes out. So the sea star has a push molecule and a pull molecule. One signal to relax and evert, another to contract and retract. That's a controlled, chemically switched deployment of an organ into the environment and back, with a separate signal for each direction. Here's the part that stopped me when I read the paper. The precursor protein that carries NGFF-yamide has, hanging off one end of it, a neurophysin domain. Neurophysin is a piece of protein architecture you might have run into before, in a completely different context. It's the carrier domain attached to vasopressin and oxytocin in our own bodies, same ancient family. In us, that lineage of signaling molecules ended up handling water balance, labor contractions, milk letdown, social bonding. The whole warm mammal package we've written 1,000 magazine articles about. In a starfish, a relative of it tells a stomach to come home. I like that more than I can defend rationally. The chemistry of holding your child and the chemistry of retracting a stomach from inside a clam share a great grandparent, anyway, mechanism. While that stomach is inside the shell, what's it actually doing? Secreting. The cardiac stomach lining releases a working mixture of digestive enzymes, proteases, which take proteins apart, amylases for carbohydrates, lipases for fats. Nothing exotic in the chemistry itself. It's a fairly standard animal digestive cocktail. What's unusual is only the address it's delivered to, and the timing is slow by our standards. A sea star working on a decent-sized mussel is looking at hours, sometimes most of a day for the whole business, from first grip to final retraction. The prey is often still alive when the stomach goes in. That's the point in the tide pool talk where somebody always winces, and it's worth being straight about it. This is a slow way to die, and there's no dressing it up. It's also completely routine on every rocky shore in the temperate world, and it's been routine for a very long time. Sea stars have been doing this since long before there were mussels shaped like modern mussels. Now, cheap here means one specific thing. Cheap in what the sea star has to build. Holding a clam shut for 6 hours is hard work by any measure. Count what the animal actually manufactures here. Enzymes, which it would need anyway. A stomach, which it would need anyway. Tube feet, which it uses for everything. That's the whole equipment list. No boring organ, no acid gland, no drill, no venom, no net. The container is free, and the container is the expensive part. And look at what that buys. A sea star's mouth is small. On a decent-sized Peasaster, it's a slot on the underside, and if the animal had to swallow its food, the mouth would set a hard ceiling on meal size. Instead, eversion breaks the link between mouth diameter and meal diameter completely. The stomach can go places the mouth cannot. Sea stars regularly eat prey wider than their own mouths, and in some cases prey with more mass than they have. If you want to see how much that matters, look at the sea stars that don't do it. There are burrowing sea stars, the sand stars, Astropecten and Luidia and their relatives, that hunt through soft sediment. They do not evert. They swallow whole. A sand star will engulf a small clam, a snail, a brittle star, take it inside, digest it internally in the ordinary way, and then spit the empty shell back out through the mouth. That works. It's clean, it's fast, and it has one hard limitation, which is that everything you eat has to fit through your mouth. Sand stars are stuck with small prey. They compensate by eating a lot of it. 2 lineages of sea star, 2 answers, and the entire difference in their diets falls out of one decision about where digestion happens. Then, there's the version of this that has to be the most underrated feeding behavior on the Pacific coast, and it belongs to a big pink animal called Pisaster brevispinus. Pink sea star, short-spined. It gets large and it lives on sand and mud flats where a lot of the food is buried. Not sitting on the surface. Buried, deep burrowing clams sit down in the sediment with only a siphon reaching up, and a sea star crawling across the top of that sand has, on the face of it, no way to reach them at all. So it digs. The animal humps up over a spot and starts moving sediment, passing bits of sand hand to hand along the tube feet, out from the mouth toward the arm tips. Pits 10 centimeters deep have been recorded, about 4 inches. The tube feet themselves stretch, and the ones near the disc can extend a long way to something like 8 inches on a large individual. And then, if it still can't reach, it sends the stomach down. Records of this behavior describe the everted stomach extending several inches down into the hole to reach a clam. The sea star's body is never going to touch. On harder ground, where digging isn't an option, the same animal will lower its stomach into a clam burrow and digest the occupant in place, down in the sediment. Picture the plumbing of that. The animal is on the surface. The meal is buried under a hand's depth of sand, inside a closed shell, and the solution is to send a length of stomach tissue down a hole, into a shell, wrap it around something alive, secrete into it, and haul the whole thing back up as a slurry. There is not a single rigid part in that entire operation. So why does this count as a real solution to the containment problem and not just a curiosity? Look at the 3 requirements again. In a buried clam, enzymes held in place. Yes, they're inside a shell, inside a burrow. There's essentially no current down there. Meal held in place? Absolutely, it's a buried clam. It isn't going anywhere. Product recovered. Also, yes, and this is the bit that makes it work. The averted stomach doubles as an absorptive surface. The tissue doing the secreting is the same tissue doing the collecting, sitting directly against the meal with almost no distance between where a molecule is released and where it gets picked up. That last point is the one I want you to carry forward, because it's going to keep coming back. In external digestion, the distance between the enzyme and the absorbing cell is the number that decides everything. Get it small, and the strategy works. Let it grow, and you're just feeding the ocean. The sea star gets that distance down to nearly zero by putting the absorbing tissue physically inside the vessel. Everything else in this video is a different way of achieving the same thing. And the first real complication comes when there's no vessel to borrow, because the meal is spread out flat across a hard surface in the open, in moving water. Which is most of a coral reef. A coral colony is a terrible place to try this. The food is real. A living stony coral is a thin skin of animal tissue, polyps, and the connecting sheet between them, spread across a hard skeleton the coral built underneath itself, protein, fat, and a huge population of symbiotic algae packed into the tissue. As a meal, it's excellent. As a container, it's nothing. It's a film on a rock, in moving water. There's no shell to slip into, no cavity, no lid. Whatever holds the enzymes here has to be brought by the animal doing the eating, and it has to work against surge, against current, and against every small mouth on the reef that would happily take a free meal in solution. This is the crown of thorns. Big animal. A grown one can run past a foot across, sometimes considerably more, with a lot of arms instead of the usual 5, and a coat of long spines. It sits on top of a coral colony. It humps up. And then it does what its relatives on the rocky shore do, with one crucial difference. There's nothing to put the stomach inside. So it puts the stomach on. The cardiac stomach comes out through the mouth and spreads across the coral surface like something poured. Flat. Wide. Following the shape of the skeleton underneath, down into the little cups where the polyps sit, into the grooves around the branches. The margins press down against the bare skeleton at the edges of the patch, and that pressed down margin is the entire technology. What the animal has built, in about a minute, out of nothing but its own tissue, is a sealed chamber. The floor is the coral. The roof and walls are stomach. The gasket is the rim, held against the skeleton by muscle and by the animal's own weight. Inside that chamber, enzymes go out, coral tissue comes apart, and the products stay in a thin film that has nowhere to go. 3 to 5 hours, according to observations of the feeding process, and the tissue is gone. Then the animal moves, and what it leaves is unmistakable. A white patch of bare skeleton with a scalloped outline that traces the shape of the arms. Clean. Not scraped, not bitten, not broken. The mineral is completely undamaged, because the mineral was never the target. The skeleton was the floor of the reaction vessel, and you don't eat the floor. Feeding scars are so distinctive that surveyors use them to tell where these animals have been without ever seeing one. You can read the shape of an absent animal off a rock. How much can a single one of them process this way? The published field numbers vary a lot, and they vary in a way that makes sense once you know what's driving it. In studies of large individuals, around 40 centimeters across, roughly 16 inches, the amount of living coral killed per day came out near 61 square centimeters in winter. In summer, that jumped to somewhere between 357 and 478 square centimeters per day. Take the summer figure, and it's a patch a bit smaller than a sheet of letter paper. Every day, from one animal with no teeth, no jaws, and no hard parts involved in feeding whatsoever. The seasonal gap is the interesting part. Warm water speeds enzymes up, Every enzyme has a temperature curve, and a cold reef in winter is running the same chemistry at a fraction of the rate. The animal hasn't changed. The reaction has. Now, that seal, a lot of what comes next is interpretation, rather than something anybody has measured with a pressure sensor under a starfish. So take it as reasoning about the physics, rather than a quoted result. The seal cannot be perfect. Coral skeleton is porous, ridged, full of pits, and a soft tissue margin pressed onto it will leave gaps at every scale, from the visible down to the microscopic. Enzymes will leak. Products will leak. So what's stopping it from leaking badly? Mucus, most likely. The everted stomach is coated in it, and mucus is one of the most quietly effective pieces of engineering in the ocean. It's a hydrated polymer mesh, and what it does to a fluid is raise the viscosity and tangle up the movement of anything trying to cross it. Small molecules still diffuse through mucus, but slowly, and bulk flow through it basically stops, which turns a leaky rim into a working gasket. Imperfect, still losing a little at the edges, and good enough for the hours the job takes. If I'm right about that, and I think the physics is hard to argue with, even if the direct measurements are thin, then mucus is doing more work in external digestion than almost anything else on the equipment list. It's the material that lets soft tissue seal against rock. And there's a strong piece of indirect evidence for how fragile the whole arrangement is, which is when these animals feed. Mostly at night. Mostly in sheltered positions, down in the base of branching colonies, on undersides, in the low-energy pockets of the reef. Feeding out on an exposed reef crest in daylight surge is not typical behavior. And during major population booms, when animals are competing for what's left, the pattern shifts, and you do see daytime feeding out in the open. Read that as a statement about the seal. A chamber made of tissue works when the water is quiet. Break the seal with surge and everything inside it. The enzymes you paid for and the meal you already liquefied goes into the water column in a second. Compare that to the sea star on the mussel, which could do its work in a washing machine and lose nothing, because its vessel had walls of calcium carbonate. That's the cost of the upgrade. You've gained the ability to eat food that isn't packaged. You've paid for it with a hard dependence on calm water. This same trick shows up in far gentler versions, and once you know the shape of it, you start noticing it everywhere. Bat stars, Pateria miniata, the flat orange and mottled ones that carpet parts of the California coast, will lay their everted stomachs over a surface and digest the film growing on it. Bacterial slime, diatoms, dead bits, whatever is settled. No prey involved at all. It's a stomach used as a mop. And the seal it needs is much less demanding because the meal is already a layer of goo stuck to a rock, and it isn't going to swim away. Cushion stars in the tropics, the Colcida group, do a version of it on coral. Different body, same logic. Sit on it, cover it, secrete, wait. And in every one of these cases, the thing to notice is that the animal has stopped borrowing a container and started manufacturing one out of itself. Every single time it eats, and only for as long as the meal takes. That's a real jump in difficulty, and it buys a real expansion of diet. The shell borrowers are stuck with prey that comes in shells. The chamber builders can eat anything flat. But look at what still hasn't been solved. Both of these strategies need the meal to be attached to something, stuck down, immobile. A mussel bolted to a rock, a coral cemented to a reef, a film of diatoms glued to a stone. Take that away, and the whole approach collapses, because the ocean's single biggest source of food floats free, suspended, drifting, individually microscopic, spread through an enormous volume of water at concentrations that would make you weep. You cannot seal a chamber around plankton. There's nothing to seal it against. So the next set of animals had to invent something a lot stranger. They build the container out in the open water, out of thread. Open water. Nothing to stand on. Nothing to press against, no floor and no wall. Somewhere in the middle of that, a small, transparent blob is hanging in the light, and around it there's a shape you can only see when the angle is right. A faint bubble of something, bigger than the animal. Ridged, chambered, with what look like windows in it. That's a house. The animal inside is a larvacean, sometimes called an appendicularian, and it's a tunicate, which makes it a distant relative of ours in a way that never stops being funny. The common shallow water one, Oikopleura dioica, has a trunk about 1 millimeter long and a tail it beats constantly. In the deep midwater there are giants, Bathochordaeus, with tails around 4 inches and houses that can span close to a yard. And nobody hides in the house. It's a filter, a whole plumbing system secreted out of the animal's own skin, inflated in the water like a tent, and then run as a machine. Here's how it's put together, and the numbers on this are the reason I wanted to get to it. Water comes in through inlet filters. Those are the coarse screens, and their pore width is somewhere around 13 micrometers, roughly the size of a red blood cell. Anything bigger than that gets excluded at the door. A coat pod, a bit of grit, a chunk of marine snow, all turned away because the animal doesn't want them, and they'd wreck the fine machinery downstream. Past the door, the water gets pushed through the food concentrating filter, and that's the part that should be famous. It's built in 3 layers. Coarse mesh in the middle, fine mesh top and bottom. And in one measured species, the pore widths of those 2 fine layers came out at about 18 hundredths and 24 hundredths of a micrometer. Say that in a more useful way. Roughly 2/10 of a micrometer, 200 nanometers. That is smaller than most bacteria. The animal is straining the ocean at a mesh size that would hold back things we needed an electron microscope to see properly. And it does it with a structure it secreted in water, in a few minutes, out of mucopolysaccharide, and a scaffold of cellulose fibers. Cellulose. In an animal, that still gets me. Tunicates are essentially the only animals that build with cellulose, the same class of material as wood and cotton, and the gene for making it appears to have arrived in their ancestry from bacteria a very long time ago. Every larvacean house drifting through the deep sea is being built with a borrowed bacterial recipe. The tail beats, water is drawn in, pushed through, and the particles that don't fit through the fine mesh pile up in a narrowing channel that ends near the mouth. And now look at the number that matters. In one careful study of animals a few millimeters long, the particle concentration inside the collecting part of the house ran between 74 and 1,089 times the concentration in the surrounding seawater. Average. Across the individuals measured, about 328 times. 300 times. That's the whole game. Because think about what open ocean water actually is from the point of view of something trying to eat. It's a nearly empty solution with a few specks in it. To get a meal out of that by chasing individual particles, you'd burn more energy swimming than you'd recover eating, and it's hopeless by orders of magnitude. The house takes that hopeless soup and turns it into something with the density of food. And here's where it connects back to the containment problem. Because larvaceans are doing something subtly different from everything we've looked at so far, and it's worth being precise about it, the house does not hold enzymes. There's no external digestion happening in there. Nothing is being dissolved. What the house does is solve requirement number 2, all by itself. In the one environment where requirement number 2 is impossible, it holds the meal. It takes food that is spread across 1,000 times too much water and puts it in one place at a workable concentration right next to a mouth. Then the animal swallows the concentrate and digests it internally, in an ordinary gut, in the ordinary way. Which sounds like a cheat, given the topic. It isn't, and here's why I'd argue it belongs in this catalog more than almost anything else. Every animal in this video is fighting the same enemy, which is dilution. The sea star's shell, the crown of thorns' seal, all of it exists to defeat dilution of the enzymes and the product. The larvation is fighting dilution one step earlier, at the level of the meal itself, and it's the only one of them attacking that problem in open water with no surface anywhere. Same war, different front. And the house has one property that none of the other vessels have, which is that it's disposable by design. Filters clog. That's not a flaw, it's just what filters do. Push seawater through a 200 nanometer mesh for a few hours, and it loads up with everything the animal couldn't take, and the flow drops, and the machine stops paying for itself. So the larvation leaves. It swims out through an escape hatch, drops the whole structure, and secretes a new one. The replacement can be inflated in minutes, from a compressed rudiment the animal has been carrying folded against its trunk the entire time, like a packed parachute. Some of them go through this cycle up to 5 times in a day. Read that as an engineering decision, and it's a bold one. The animal has chosen to throw away its most complex structure several times daily rather than maintain it. I've gone back and forth on whether that's expensive or cheap. It's clearly expensive in materials, and larvations put a serious fraction of their carbon budget into house production. But maintenance has costs too, and cleaning a 200 nanometer mesh in the middle of the ocean, with no hands, is not obviously possible at any price. Sometimes the right answer really is to throw it out and print a new one. There's an aftermath to this that got a lot of attention when the deep water version was finally filmed properly. Researchers at the Monterey Bay Aquarium Research Institute developed an instrument to image these things in place, in the deep mid-water, without destroying them, which had been the problem for decades. Larvation houses are so fragile that any net that catches one turns it into slime. What they found is that the discarded houses of giant larvations, loaded with everything the filter caught, sink. They fall out of the sunlit water carrying carbon down with them, and they do it fast because a clogged house is dense. A used vessel full of food going to the seafloor, which is where a whole other set of animals is waiting, and we'll get to them, because some of the strangest chemistry in this entire subject happens down there on the stuff that falls. For now, keep the shape of the idea. An animal built a container out in open water where there was nothing to build against and used it to concentrate a meal 300-fold. The larvation throws its container away when it's full. The next group eats it, down in the sediment, in a tube shaped like a horseshoe with both ends open to the water. There's a worm doing something that looks like industrial process design. Chaetopterus, the parchment tube worm. If you've dug one up, you'll remember the tube more than the animal, because the tube is a tough, papery cylinder that goes down into the mud and comes back up, and the worm lives in the bend of it. The body is bizarre, even by polychaete standards. Segments that don't match each other, a front end with flattened wings, a middle section carrying 3 big fans, which are modified body appendages, and those fans beat continuously to drive a current through the tube, water in one chimney, out the other. And stretched across the middle of that current, held open like a windsock, is a bag made of mucus. The worm secretes it. 2 of those winged appendages up front produce the material and hold the mouth of the bag open, and water gets pumped straight into it. The structure of that bag is where this stops being cute and starts being impressive. Put it under a microscope, and you find weave. 2 crossing arrays of parallel filaments, one set spaced at about 3/4 of a micrometer, the other at just under half a micrometer, laid across each other. An actual mesh made of slime by a worm with no hands. That geometry means the bag holds back particles down to around half a micrometer and lets most of the smaller stuff pass, which puts it in bacterial territory, and it means the worm is filtering out things that are, to any normal way of thinking, invisible. Now the part I find properly clever. The bag fills up. Same problem the larvation has. Same physics. Same inevitable clogging. And Chaetopterus has a structure sitting behind the bag, a little cup. And as the bag loads with material, the worm rolls the whole thing forward into that cup and balls it up. Mucus, plankton, bacteria, detritus, everything. Wound into a single pellet. Then a ciliated groove running along the worm's back carries that pellet forward, hand over hand, to the mouth. And the worm eats it. All of it. The catch and the net together. That is the cleanest answer to requirement number 3 that we're going to see in this entire video, and it's worth stopping on. Every other animal here has to solve the problem of separating the product from the container. The sea star has to leave the shell behind. The crown of thorns has to lift off the skeleton. The larvation throws its house away. Chaetopterus never has that problem because its container is food. Mucus is glycoprotein. Sugar chains hung on a protein backbone. It's a perfectly reasonable thing to digest, so when the worm swallows the loaded bag, the vessel and the contents go through the same gut and come out as the same nutrition. Nothing is discarded. Nothing has to be pried loose. There's no recovery step at all because the recovery step was designed out of the system. Then a fresh bag gets secreted, and it starts again. Over and over, all day, all night, for as long as the current holds. Little extra detail on this animal that has nothing to do with feeding and that I'm including anyway. Chaetopterus glows. Disturb 1 and the mucus it produces luminesces blue. It's one of the few bioluminescent annelids, and nobody has fully nailed down what the glow is for. A worm that lives its whole life buried in mud, making light. The same net and swallow logic shows up in a completely unrelated animal. And once you see it in 2 places, you realize the ocean has invented it more than once. Vermitid snails. These are gastropods that have given up on being snails in the way you'd recognize. They cement themselves to rock as juveniles, and then grow their shell as an irregular tube stuck to the substrate, looking more like a worm's home than a snail's. They never move again. A sessile snail has an obvious problem, which is that it can't go find food, so it fishes. The animal produces mucus from a gland in its foot and casts it out into the water as a net. Some species put out a sheet many times the size of their own body, drifting downstream from the tube opening, catching whatever the current carries. Plankton, particles, bits of organic material, and then it reels it in. The radula, which in most snails is a rasping tongue for scraping algae off rocks, gets used here as a retrieval tool, hauling the loaded mucus back into the tube. Net and catch swallowed together, exactly like the worm. 2 animals from 2 phyla that split apart more than 500 million years ago, both landing on the same solution. Build a disposable container out of edible material, fill it with the ocean, then eat the container. Now, is this free? No, nothing is free. Mucus costs protein. Mucins are big, heavily modified proteins, and the modification alone is metabolically demanding. An animal that produces a new net every few minutes is running a continuous protein export operation out of its own tissues, and if the current stops bringing food, that export becomes a straight loss. There's a reason net feeders live where currents are dependable. Look at where Chaetopterus tubes cluster and where vermitids cement themselves, and it's the places with reliable water movement. The strategy is a bet that the ocean will keep delivering, and it's a bet you have to keep paying to stay in. But notice what the mucus buys physically, because this material is doing more work than it gets credit for. Mucus is a polymer network that's mostly water, which makes it cheap in dry mass for the volume it occupies. It's sticky, so particles that touch it stay touched. It's flexible, so it deforms with the current instead of tearing. And when you're done with it, it's digestible. Try to design a filter with that combination out of anything else. Rigid materials break and surge. Anything strong enough to survive is too expensive to throw away. Anything cheap enough to discard usually isn't fine enough to catch what matters. Slime wins on every axis at once, and it wins so thoroughly that it shows up independently in worms, snails, tunicates, and, as we'll see shortly, in the largest fishing structures any animal builds in open water, blue water, a long way from any bottom. Something is hanging in the light and it takes a second to work out what you're looking at because most of it is invisible. There's an animal in the middle, soft, gelatinous, maybe 2 inches across, with a broad muscular plate it uses to swim. A pelagic snail. One of the sea butterflies, though this group has given up on anything you'd call a shell. And spread above it, catching the light only at the edges, is a sheet of mucus that can reach about 2 yards across. Gleba cordata, Corolla spectabilis. Same trick, related animals. And the numbers on it are worth saying out loud because they sound made up. The animal is roughly 50 millimeters, 2 inches. The web can hit about 2 meters, 6.5 feet. Run that ratio through a human body and you get a person casting a net 80 yards wide. Not carrying it, not anchoring it, casting it out into the air, unsupported, from glands in their own skin, and then hanging underneath it. The mucus comes out of glands running along the border of that swimming plate. The web forms above the animal, and then the animal stops swimming and starts sinking. Slowly. Falling through the water with an enormous sheet of slime stretched out overhead, letting the water column pass through it. Anything in that water that hits the mucus sticks. Phytoplankton, small zooplankton, organic particles. The web has pores in it, and measurements put most of them above 15 micrometers, which sets a rough lower bound on what it bothers to keep. And then the collection. Cilia. The whole web gets moved, steadily, down toward the mouth by ciliary action, hauled in like a sail being furled by a million tiny hands, with the catch still stuck to it. Web and food together, into the animal. Same design as the worm in the mud, at a scale about 1,000 times larger, in an animal that has nothing to stand on. The physics of how that sheet holds together is worth a moment, because at first glance it should be impossible. There's no frame, no spokes, no radial threads, nothing under tension. A structure 2 yards across with no skeleton, out in the open ocean. 2 things are working in its favor. The mucus is mostly water, so it's close to neutrally buoyant. It doesn't have to be held up, because it isn't really falling relative to the water around it. And at that scale, in that fluid, the ocean is a much stiller place than our intuition suggests. Away from surf and current shear, the water in a patch a couple of yards across mostly moves as a piece. There's no wind. There are no gusts. The web doesn't have to survive being flapped around because nothing is flapping it, which is also its whole vulnerability. Disturb the animal and it drops the entire structure and swims, abandoning the web and everything caught in it. A single startled fish costs it the whole meal. I have a slight soft spot for that. A 50-millimeter animal that has invested a serious portion of its energy budget into a 6-foot fishing apparatus made of its own body fluid, and its emergency plan is to let go and run. So let's take stock of the net feeders as a group, because they've done something specific, and it's easy to lose track of what. None of these animals is digesting anything outside its body. Not the larvacean, not the tube worm, not the vermitid, not the sea butterfly. There are no external enzymes anywhere in this part of the story. What they've solved is requirement number 2, in the hardest possible setting. They hold the meal in a place where nothing can be held, and the way they do it is by moving the boundary of their own body outward, into the water, in the form of a structure that's part of them without being made of cells. That's a real philosophical oddity if you poke at it. Where does a larvacean stop? The house is secreted, shaped, maintained, and operated by the animal. It does a job that in you and me is done by organs, and the animal dies without it. It isn't alive. It also isn't the environment. Biologists sometimes call structures like that extended phenotypes, beaver dams, spider webs, caddisfly cases. The larvation house belongs on that list, and it's the strangest member of it, because it's a filter with a mesh finer than most cells, built by an animal you could lose on your fingertip. And every one of them shares the same weakness, which is that they only work on food that's already loose in the water, suspended, small, free-floating. Now change the problem. Take the food and lock it inside a mineral. Not a shell you can pry open, not a coral skeleton you can lay a stomach across. Actual mineral, with the nutrition distributed inside it, chemically bound into a matrix of calcium phosphate crystals and protein fiber, sealed in a structure that is one of the hardest things a vertebrate body makes: bone. A whale dies, and eventually the carcass comes down. Everything soft goes first, and it goes fast. Sleeper sharks, hagfish, amphipods, crabs, an entire scavenging economy that can strip a large whale carcass in a matter of months. Then that stops, and what's left on the seafloor is a skeleton. And a whale skeleton is not an empty object. Whalebone is loaded with lipid. Reviews of whalebone composition put the fat content of some vertebrae at a very substantial fraction of the bone, with reported values in parts of the spinal column running into the range of 40 to 55% by weight in the tail region. There's also collagen, which is protein wound through the mineral, so there's a fortune sitting on the seafloor. Concentrated, dense, and completely inaccessible, because every calorie of it is inside a rock. You cannot bite it. Nothing at that depth has jaws that would help. You cannot net it. You cannot seal a stomach over it and expect enzymes to do anything, because enzymes bounce off hydroxyapatite. Proteases do not dissolve mineral. To get into a bone, you need a different kind of chemistry entirely, and you need to apply it to a solid surface, underwater, for months. The animal that figured this out was only described in 2002, and it doesn't have a mouth. 2002, Monterey Bay. A research team was out looking for clams. That's the honest version of the story. Bob Vrijenhoek at the Monterey Bay Aquarium Research Institute had a cruise running with a remotely operated vehicle, working the canyon. And the target was deep-sea clams. Routine work. And then the camera came over a gray whale carcass on the bottom, at a depth of about 2,900 meters. Call it 9,500 feet, nearly 2 miles down. The soft tissue was long gone. What was left was bone lying in the sediment, and the bone was covered in something red. Plumes. Thousands of small, feathery red structures standing up off the surface of the vertebrae, waving in whatever slow current existed down there, so dense in places that the bone underneath was hard to see. They pulled up samples. Greg Rouse, a polychaete specialist, got a look at them, and the animals turned out to be worms in the family Ceboglinidae, related to the giant tube worms of hydrothermal vents. The genus got named Osedax, which is Latin, and which translates roughly to bone devourer. Then the dissection started, and the anatomy is where this becomes one of the strangest animals in the ocean. Start at the top. The red plume is a crown of palps sticking up into the water. That's the respiratory surface. It's red for the same reason your blood is red, hemoglobin, and it's doing gas exchange, pulling oxygen out of the seawater. Below that, a trunk sitting in a clear tube of mucus attached to the bone surface. And below that, going down into the bone, the part nobody expected. Roots. Actual branching root tissue, green or pink or yellowish, depending on the species, ramifying through the interior of the bone the way a plant root ramifies through soil. Not a burrow with a worm in it. Tissue, grown into the mineral, following the internal spaces, spreading out into a network that can occupy a substantial volume of the bone. Now, for the part that stops people, there's no mouth. And I mean none at all. The adult female Osedax has no mouth, no gut, no stomach, no intestine, and no anus. The entire digestive tract is absent. It was never built. An animal is sitting on a pile of the richest food in the deep sea, and it has no opening through which food could enter. The males make it stranger. Osedax males are microscopic. They never develop past what is essentially a larval stage. They never feed at all. And they live inside the female's tube. Not one of them. Between 50 and 100 of them in a single female, in some species, a whole crowd of arrested larvae, living off the yolk they hatched with, present for one purpose. So you have a female with no digestive system, hosting a harem of dwarf males who also have no digestive system, growing roots into a whale's spine. If you were designing an animal to make people ask how, this would be it. Since that first find, the group has turned out to be everywhere. Dozens of species, described from whale falls all over the world, at a wide range of depths, from thousands of meters down to shallow water in some cases. Researchers have put out experimental bone deployments and had Osedax colonize them. And the animals aren't strictly whale specialists either. They'll take other vertebrate bone, fish bone. In experiments, cow bone dropped into the deep sea has been colonized, which means that every time a large vertebrate has died at sea for a very long time, something like this has probably been waiting for it. Now to the actual problem, because I don't want to skip past how hard it is. Bone is a composite material, and composites are hard to attack precisely because they're composites. There are two phases in there doing two different jobs. Phase 1 is the mineral. Hydroxyapatite, a crystalline calcium phosphate laid down in tiny plates. That's what makes bone stiff and what makes it resist compression. Phase 2 is the organic part. Mostly collagen, a fibrous protein, wound through the mineral. That's what makes bone tough instead of brittle. And it's also the part with nutritional value, along with the lipid that's stored in the spaces. Digestive enzymes only work on phase 2. A protease will happily take collagen apart. Collagen is a protein and proteases eat proteins. That part is easy. The obstacle is reach. The collagen is embedded in the mineral, coated in it, locked in a crystalline matrix, and an enzyme molecule arriving at the surface of a piece of bone finds mostly calcium phosphate in its way. Proteases do not dissolve rock. They bounce. Same for the fat. Lipid stored in the interior spaces of a bone is behind a mineral wall, so the sequence has to be first remove the mineral, then digest what's underneath. In that order. There's no way around it and no shortcut. Removing the mineral means dissolving hydroxyapatite, and hydroxyapatite dissolves an acid. That's it. That's the only practical chemistry available to a soft-bodied animal at 2 miles depth, which means Osedax has to do something that almost nothing else in the animal kingdom attempts. It has to generate acid, hold it against a solid mineral surface, in seawater, and keep doing it for months. And seawater is the worst possible medium for that plan, because seawater is buffered. The ocean is a carbonate buffer system. It resists changes in acidity, deliberately and effectively. And that's the same chemistry that makes ocean acidification such a slow, grinding process rather than a sudden flip. Dump acid into seawater and the buffer soaks it up. So the worm is trying to run an acid attack inside a solution specifically designed by geology to neutralize acid attacks. The way it manages that is one of the best pieces of convergent evolution I know of, and it uses a molecular machine you have in your own body, doing almost exactly the same job. The machine is called a vacuolar proton pump. In the literature, you'll see it written as VATPase, or VHA. It's a rotary motor, physically a rotary motor, sitting in a membrane with a spinning part and a stationary part, burning ATP to turn a rotor that shoves hydrogen ions from one side of the membrane to the other. Every complex cell on Earth has these. Yours are running right now by the million, and normally they point inward. That's the thing to hold in your head. In a typical cell, proton pumps sit in the membranes of internal compartments, and they acidify the inside of those compartments. Your lysosomes, the little acid bags where cells break things down, are acidic because VATPases pump protons into them. The chemistry stays safely locked in a bubble inside the cell. In 2013, a team led by Martin Trigueros, working with Sigrid Katz and Greg Rouse, published a study asking a simple question: how does Osidax get into bone? They looked at the root tissue. The roots express proton pumps in extraordinary quantity, not the ordinary background level every cell has. Extremely high amounts, enough that the tissue lights up under antibody staining like it's been switched on. And critically, the pumps aren't only in internal vesicles. They're in the apical membrane, the outward-facing surface, the side of the cell that touches the bone. The worm has taken the machine that acidifies the inside of a cell and mounted it facing out. Alongside it, the same tissue expresses carbonic anhydrase. And that enzyme is the supply line. Carbonic anhydrase takes carbon dioxide and water and converts them into a bicarbonate ion and a free proton, fast. Some forms of it are among the quickest enzymes known, turning over on the order of 1 million reactions per second in a single molecule. Carbon dioxide is the one thing an animal always has, because metabolism produces it constantly. So the worm is taking its own waste gas, running it through carbonic anhydrase to strip protons off it, and feeding those protons to the pumps in the membrane facing the whale's bone. The result is acid, delivered directly onto hydroxyapatite. The mineral dissolves. Calcium and phosphate go into solution, and the collagen and lipid that were locked inside are suddenly loose. Now here's the convergence, and it's the good part. You have cells that do this. Right now, they're called osteoclasts, and they're the demolition crew of your skeleton. Bone is a living tissue that gets constantly rebuilt, and osteoclasts are the cells that take the old material away. The way they do it is this. An osteoclast crawls onto a bone surface. It forms a ring of extremely tight adhesion around a patch, a structure called the sealing zone, which clamps the cell membrane down against the mineral all the way around. That ring creates a sealed pocket between the cell and the bone, a microscopic chamber. Then, into that chamber, the cell pumps protons with VATPases in its membrane, using carbonic anhydrase to generate them, and the pocket goes acidic. Down around pH 4.5 in an environment where the rest of your body sits near 7.4. The mineral in that patch dissolves. Then the cell releases enzymes, including one called cathepsin K, which takes apart the collagen that's been exposed. Look at what your osteoclast is doing. In the language we've been using this whole time, it seals a chamber against a solid surface. It holds its chemistry inside that chamber at a working concentration. It dissolves the container, then digests the contents, then absorbs the product across the same membrane, doing the secreting. That's the containment problem, solved at the scale of a single cell in your femur. And a marine worm 2 miles down in the Pacific, in a lineage that split from ours before there were vertebrates at all, has arrived at the same solution with the same molecular parts. Nobody copied anybody. There are only so many ways to dissolve a calcium phosphate crystal, and both lineages found the same one. Osidax has one advantage over the osteoclast that I think is underappreciated. It doesn't have to make a seal. Remember the crown of thorns pressing a stomach margin against rough coral, and how that seal is only as good as the water is calm. The osteoclast has the same difficulty at a smaller scale, and it solves it with that ring of adhesion. The worm sidesteps it entirely because the roots grow into the bone. The tissue sits inside a cavity, And the cavity is exactly the shape of the tissue because the tissue made it by dissolving its way in. There's no gap for anything to leak out of. The mineral is the vessel, and the worm is inside the vessel. And the seal is automatic. And it stays automatic as the root advances, because the root is always filling the hole it just made. That's why this works in seawater despite the buffering. The acid never meets the ocean. It's released into a space that has no meaningful connection to the outside water. Against a surface, it's dissolving, and the reaction runs to completion in a volume you'd need a microscope to see. Then the recovery, and here the picture gets more interesting and slightly more contested. The freed material has to become worm. The roots are packed with bacteria, symbionts of the order Oceanospirillales, living inside specialized host cells beneath the skin of the root. Those bacteria are heterotrophs, they metabolize organic material, and the worm draws nutrition from them, which is the standard interpretation, and it's well supported. The open question has been how much digestion the worm does for itself. Transcriptome work on one species, Osedax japonicus, found the worm's own root tissue expressing genes for enzymes that break down collagen, along with machinery for taking material into cells directly, which suggests the worm isn't just a passive landlord waiting for its tenants to hand over lunch. It appears to be running part of the digestion itself and absorbing the products through the root epithelium. The honest summary is that both are probably happening, and the balance likely varies between species. It's an active area, and anyone who tells you it's settled is ahead of the evidence. What isn't in doubt is the timescale. This is slow. A whale skeleton can support these animals for years. There's no rush at 9,000 feet, and there's nothing else competing for what's inside the bone, because nothing else can get in. That's the reward for solving the hardest containment problem in this video so far. You get a resource that is, functionally, yours alone. And Osedax isn't the only animal that took the acid route. Once you know what to look for, you find the same strategy applied to a different mineral, by animals that couldn't be less related to it. Pick up an old oyster shell from a bay somewhere on the Atlantic coast and look at the outside of it. If the shell has been in the water long enough, you'll often find it pocked with small round holes, a couple of millimeters across, sometimes hundreds of them. And if you break the shell open, the inside is honeycombed. Tunnels and chambers running through material that's supposed to be solid, and in a fresh shell, the holes have yellow or orange tissue sitting in them. That's Cleona, a boring sponge. Sponges are the animals we usually file under harmless. No nerves, no muscles, no organs, filtering water and minding their own business. This one carves through calcium carbonate. The obvious guess about how it works is wrong, and the way it's actually done is much stranger. You'd assume scraping, some abrasive surface wearing the shell down over time. A sponge has nothing to scrape with. No hard parts capable of it, no muscles to press with, nothing that could function as a tool. What it has is a specific type of amoeba-like cell called an etching cell, and what an etching cell does is hard to picture the first time you read it. The cell crawls onto the carbonate surface. It extends thin fingers of cytoplasm out and around a small patch of the shell, and then it chemically cuts a groove, following the outline of that patch, working around it and underneath it. It's cutting out a chip, like a hole punch made of cell. The chemistry that does the cutting is acid, applied locally in the tiny space between the cell membrane and the mineral, and the mechanism involves the same class of machinery we just met in the worm and in your own skeleton. Proton transport, local pH control, right at the interface where the cell touches the rock. Then the groove closes underneath, and the chip comes free. And the chips are the giveaway, because they have a characteristic shape and a characteristic size, scalloped little fragments, tens of micrometers across, all of them cut by the same process to roughly the same specification. They get swept out of the sponge through its water canals and expelled. Sediment full of those distinctive chips is one of the ways you identify boring sponge activity without ever seeing the sponge. Most of the carbonate that leaves a shell this way leaves as chips. Only a portion of it is actually dissolved, which is a smart division of labor if you think about the energy budget. Dissolving mineral is expensive because you have to pay for every proton. Cutting a groove around a chip and letting the chip fall out means you only pay for the outline instead of the whole area. The sponge is minimizing its acid bill by cutting rather than melting. Now, an important honesty check, because it would be easy to slide this into the story as another feeding strategy, and it isn't one. The boring sponge is not eating the shell. There's no nutrition in calcium carbonate. Cliona feeds the way sponges feed, by pumping water through itself and capturing bacteria and fine particles. The excavation is about real estate. It's building a home inside a hard object where nothing can reach it. And the tunnel system doubles as its plumbing. Same molecular machinery as Osedax. Same problem of applying acid to a mineral underwater. Completely different reason for doing it. I think that's more revealing than if it had been another digestion story. It tells you that the acid against a surface toolkit is generic. Evolution has built it repeatedly, out of parts that were already lying around in every cell, and then pointed it at whatever needed dissolving. Food, in the worm. Housing, in the sponge. Your own skeleton, in the osteoclast. The tool is old. The application is whatever the animal happens to need. And there's a third group that took the same toolkit and used it to get through a wall. Predatory snails. If you've ever collected shells, you've held the evidence. A small clam or mussel shell with one perfectly round hole in it, usually near the middle, often countersunk like it was made with a proper drill bit, clean edges, tapered walls. That hole was made by another snail eating the animal inside. Two big groups do it. The moon snails, which are the smooth globular ones that plow through sand flats leaving those wide furrows, and the murexes and dog whelks, which are the knobbly ones on rocky shores. Both have a structure called the accessory boring organ. In the moon snails, it sits on the proboscis. In the whelks, it sits on the sole of the foot. Either way, it's a specialized patch of secretory tissue whose job is to soften calcium carbonate chemically, and then the snail alternates. Apply the organ. Chemically weaken a small patch of shell. Pull back. Bring in the radula, which is the toothed ribbon most snails use for scraping algae, and rasp away the softened material. Apply the organ again, rasp again. Chemical, mechanical, chemical, mechanical, for hours, sometimes for more than a day on a thick shell. That alternation is doing something specific and clever. Pure rasping on unsoftened shell would wear the radula out and get nowhere. Pure dissolution would cost an enormous amount of acid because you'd have to dissolve the entire volume of the whole. Softening a thin layer and then scraping it away means the chemistry only has to weaken the surface, and the teeth do the removal. Same logic as the sponge cutting chips instead of melting. Pay for as little dissolution as you can get away with. When the hole finally goes through, the snail puts its proboscis in, and now look at what it has. A sealed calcium carbonate box with a single small opening in it, containing a meal that cannot get out, and a soft, flexible feeding tube inserted through that opening. That's the sea star's solution again. The prey's own shell as the reaction vessel. The difference is that the snail didn't wait for the shell to open. It made its own door. One last thing about these holes, because it's one of the few places in biology where you can put a date on a behavior. Drill holes fossilize. The hole is a permanent feature of a shell. It has a recognizable geometry, and shells preserve well. So the record of one animal drilling into another is written into rock all over the world, and boreholes that look very much like these turn up in shelly fossils from more than 500 million years ago. Most behavior leaves nothing behind. This one leaves a signature you can measure with calipers. Now to an animal that solved this before there were animals with two ends. Watch an anemone take a fish. The tentacles fire, the fish stops moving, and the tentacles fold inward and pass it toward the center of the disc. The mouth opens, which on a big anemone is a slot that can stretch to a size you would not believe from looking at it closed. And the fish goes in. Then the mouth shuts, and the animal sits there looking like a lump. What's happening inside is where the cnidarians get interesting, because their body plan should not work at all. A sea anemone is structurally a bag with one hole. And that's the literal anatomy, straight off a dissection table. There's a mouth, it leads to a cavity, and the cavity is closed. No stomach, then intestine, then exit. One opening, doing both jobs. The technical name for the space is the gastrovascular cavity, and the name is telling you that it's the gut and the circulatory system at the same time, because there isn't a separate one. You and I run a tube, food goes in one end, moves in one direction, gets worked on by different chemistry in different compartments, and leaves at the other end. That design lets you specialize each section, and it lets you eat continuously. A one-hole animal can't do any of that. It's a batch reactor. Load it, close the door, run the reaction, open the door, dump the residue, load the next one. And a batch reactor with a hole in the top has an obvious flaw: the hole. The first thing to notice is that they close it properly. There's a muscular pharynx leading down from the mouth, and it can be pulled shut, and the whole oral disc can be drawn in until there's no opening at all. An anemone that's feeding, or one that's just been poked, will pull itself into a closed blob with no visible mouth. That's a lid. And a lid on a bag turns the whole animal into a reaction vessel with the meal inside it. But the cavity is not just an empty bag, and the internal architecture is the part that makes the whole strategy work. Run vertical sheets of tissue inward from the body wall, like the partitions in a grapefruit. Those are the mesenteries. They do several jobs at once. They multiply the surface area of the lining enormously. They carry muscle, and they carry gonads. And along the free inner edge of each mesentery, where it hangs into the cavity, the tissue is thickened into a cord. Those cords are the mesenterial filaments, and they are the business end of the entire system. Pack into one of those filaments the following: gland cells that secrete digestive enzymes, cilia to move fluid and particles along it, and stinging cells, cnidocytes, the same category of cell the animal uses to catch prey in the first place. The result is a rope of digestive tissue, armed, mobile, and able to be draped wherever it's needed. Think about how different that is from your gut. Your digestive surface is a wall. It's the inside of a tube. It stays where it is, and the food moves past it. The anemone's digestive surface can go to the food. If the fish is large, the filaments wrap it. If part of the meal is tucked into a corner of the cavity, filaments reach into the corner. The animal is applying its digestive tissue directly onto the specific piece of material it wants dissolved, at whatever angle the geometry demands, which means the distance between the enzyme and the target is nearly zero everywhere. That's the number that matters. The same number that made the sea star's everted stomach work. Distance between secretion and target. The cnidarians got it to zero by putting the digestive tissue on flexible cords and letting them go find the food. Now the two-stage part, because cnidarian digestion has a step that ours mostly doesn't. Stage 1 is extracellular, and it happens in the cavity. Enzymes go out. The meal comes apart into fragments and soluble material, and after a few hours a fish is reduced to a slurry with some bone and scale in it. Stage 2 is intracellular. The cells lining the cavity, the gastrodermis, take those fragments into themselves, physically engulf them, the way an amoeba engulfs a particle. package them into internal bubbles, and finish the digestion inside the cell, in a compartment acidified by, yes, the same proton pumps we met in the whalebone. So a canidarian digests a fish twice. Once in the open cavity, coarsely, and then again inside individual cells, precisely, which sounds redundant, and it isn't, and the reason it isn't goes right back to the containment problem. Extracellular digestion in a bag has a recovery problem. You've got a soup of amino acids and sugars in a cavity that has to open eventually to dump the bones out. Every time that mouth opens, whatever is still in solution is available to the ocean. Intracellular digestion has no recovery problem at all, because the products appear inside the cell that needs them. There's no gap to cross, nothing to lose. So the two-stage system is a compromise between speed and security. Break the meal down quickly and roughly in the open cavity, then get the pieces inside cells as fast as possible, and do the valuable part where nothing can escape. Cnidarians have been running that arrangement for something over 500 million years. It works well enough that anemones eat fish, corals build reefs off it, and jellyfish do it while drifting. And then some of them took the next step, which is the one that actually belongs in this video. They started sending those filaments out of the animal entirely. Poke an anemone of the right sort and it fights back with threads. They come pouring out, white or pink, fine, sticky, unspooling into the water in a tangle, sometimes through the mouth, sometimes straight through little pores in the side of the body wall that exists specifically for this. Anyone who has kept a saltwater tank with the wrong anemone in it knows the experience and knows that whatever those threads touch has a bad day. Those are acantia. They're the free ends of the mesenterial filaments, extended down past the mesentery, hanging loose in the cavity until they're needed. Same construction as the filaments, gland cells, cilia, stinging cells, and they can be fired out of the animal. Everybody has always classified them as defense, and that's clearly one function. In 2008, a study out of Tel Aviv University by Amy Schlesinger and colleagues, published in the Proceedings of the Royal Society, put a different question to them. The setup was straightforward. They fed sea bream larvae to a small anemone, Aeptasia diaphana, and watched what happened after the fish went in. The expectation was that the tentacles do the killing and the cavity does the digesting, which is what the textbooks say. What they saw was that the acantia went to work on the swallowed fish, wrapped it, and kept stinging it inside the animal after it was already caught. Nematocysts firing into a fish that had already been swallowed, which sounds like overkill until you look at what came out of those stings. At least 5 digestive enzymes delivered into the fish's body, including ones that go after fats and ones that go after blood cells. The stinging apparatus wasn't only injecting venom, it was injecting digestive chemistry through a needle into the tissue it wanted broken down. Then they did the experiment that makes the whole thing land. They took acantia off the anemone, detached them, put those loose threads in a dish with fish larvae, with no anemone anywhere in the arrangement. The threads killed the fish, and then started disintegrating them. A piece of tissue, cut off from the animal it belongs to, sitting in a plastic dish, successfully hunting and dissolving prey on its own. I've read a lot of feeding physiology, and that one still lands as strange. It's a freestanding digestive organ. It doesn't need the rest of the anemone to function. And once you take that seriously, the obvious next question is why keep it inside at all? They don't, always. There's a behavior called extracolinteric feeding, and it's exactly what the word says. Outside the colinteron, outside the cavity. Corals do it routinely. A polyp encounters something edible sitting on the substrate next to it, something too big or too awkward to get through the mouth, and instead of trying to swallow it, the polyp pushes mesenterial filaments out. Through the mouth, or straight through the body wall and lays them over the food. The digestive tissue leaves the animal and goes to the meal. That is the same move the sea star makes with its stomach, arrived at by a completely different body plan. In an animal with no stomach to evert, the cyanidarian doesn't have one organ to send out. It has dozens of cords, each of which is independently deployable, each armed, each secreting. And the containment logic is the same as everything else in this video. The filament gets laid onto the food, wrapped around it, pressed against it, with mucus between, so that the volume the enzymes are working in is a film of fluid a fraction of a millimeter thick, trapped between two surfaces. Then, the recovery. The filaments are ciliated along their length, and those cilia beat in a direction, and the products of digestion get moved along the filament, back toward the polyp, and in. The same structure is the applicator and the conveyor belt. It delivers the chemistry outward and hauls the results back. Corals also apply those filaments to things that are not food, in disputes over space with their neighbors, which is a whole other subject and a different video. What matters here is the equipment. The same cord does both jobs, because dissolving tissue is dissolving tissue. Now the version that broke my brain a little when the paper came out. Cassiopeia, the upside-down jellyfish, the ones in mangrove lagoons in Florida and the Caribbean that lie on the bottom with their bells down and their oral arms up, farming symbiotic algae in their tissues like a coral would. For years, people who swam in those lagoons reported something they called stinging water, not a jellyfish sting. You'd get an itching, burning sensation without touching anything. It was widely assumed to be broken off bits of tentacle drifting around or possibly tiny larvae of something else. In 2020, a team led by Cheryl Ames published the answer in Communications Biology, and the answer was better than any of the guesses. The jellyfish releases structures into its mucus. The team named them cassiosomes. A cassiosome is a small blob, and its outer surface is made of stinging cells. Inside it, there's a core containing the jellyfish's symbiotic algae, and on the outside, cilia, which means it swims. These things leave the animal, travel through the water under their own power, and remain active for up to 10 days. In the lab, cassiosomes released into water with brine shrimp killed them. So the jellyfish seeds the water around itself with self-propelled, autonomous, stinging units, and letting them do the killing at a distance. Detached weapons, made of the animal's own cells, swimming. Now the limits of that claim matter. What was demonstrated is that these structures sting and kill. How much digestion happens out there in the water, and how much of the resulting nutrition ever makes it back to the jellyfish, is a long way from settled, and the paper doesn't pretend otherwise. The likeliest payoff is prey that dies nearby and drifts down onto the animal, which in a shallow mangrove lagoon is a reasonable bet to make. Still, look at the trajectory across this one group of animals. Digestive tissue lining a closed cavity, then digestive tissue on mobile cords inside that cavity, then those cords pushed out through the mouth onto food outside. Then armed cellular packages released into the open ocean to operate independently for over a week. Each step moves the chemistry further from the body. Each step gives up a little more control in exchange for reach. And the reason cyanidarians could push that far when the sea star couldn't comes down to one structural fact. They fire their chemistry through needles. A nematocyst is a syringe. It's a capsule under enormous internal pressure with a coiled thread inside it, and when it triggers, the thread turns inside out and drives into tissue faster than almost any movement in biology. Anything delivered that way is delivered inside the prey, not into the water next to the prey. Inside it, past the skin, into a volume that the ocean cannot reach, which is the ultimate answer to the dilution problem, and it's the same answer the last predator in this catalog arrived at, with a very different piece of hardware. Before that, though, there's a case where the food fights back with a material nobody expected, and where the containment problem gets solved by moving in the opposite direction, inward, all the way down to the inside of a single cell, cold water. British Columbia, maybe Southeast Alaska, deep enough that everything is blue and gray. There's a sponge on the rock, and it's not the soft kind. This one is a glass sponge, and the name is literal. Its skeleton is silica, actual glass, laid down as spicules with 6 rays. And in this group, the spicules can fuse into a rigid lattice, so the whole animal is a scaffold of welded glass with living tissue draped through it. They're beautiful, and they're ancient, and in the deep waters of the Pacific Northwest they build reefs, which is something people assumed had stopped happening 100 million years ago until somebody found live ones in the 1980s. And crawling across this one is a sea slug. Big, pale, warty, unhurried. Adorid nudibranch Peltodorus lentiginosa or Archodorus oedneri, depending on which one you found, and both of them are documented predators of glass sponges. Which raises a question that I don't think gets asked often enough. How do you eat glass? Let's be specific about the problem, because it's worse than it sounds. A sponge is a composite all the way through. Living cells distributed through a 3-dimensional mesh of mineral needles, with the mineral often making up the great majority of the dry weight. In a glass sponge, the framework is silica, and the framework is most of the animal. The nutrition is the thin part. Cells tucked into the spaces of a structure that isn't food. Add the chemistry. Sponges are famously loaded with secondary metabolites, defensive compounds that make them unpalatable or outright toxic to most things. There's a whole pharmaceutical industry that goes prospecting in sponges for that reason. So the meal is glass, laced with poison, with the food distributed through it like raisins in a wire sculpture. Now try to apply anything we've learned so far, seal a chamber over it and secrete. You can't get a seal. The surface is a mesh of needles. A soft tissue margin pressed onto that geometry leaks in every direction, and it also gets punctured. Dissolve the skeleton first, the way the worm does with bone. Silica doesn't behave like calcium phosphate. Acid is the wrong tool here. Dissolving silica is a slow process that goes better in alkaline conditions, and no digestive system in the world runs on that chemistry. Break it up mechanically? With what? A sea slug has a radula, and a radula is a ribbon of small, chitinous teeth. Chitin against glass is not a contest anybody wins, so the nudibranch has 3 options, and 2 of them are closed. What it actually does is one of the most instructive answers in this whole catalog, and I say that because it goes in the opposite direction from everything else here. It swallows the glass. Study the feces of a sponge eating Dorid, and you get the answer straight away. Work on Doris verrucosa. Feeding on a sponge found that roughly 97% of the spicules the slug ingested came back out again, mostly unbroken. In some cases, the spicules were still arranged the way they'd been arranged inside the sponge, holding their original structure as they passed through the animal. The slug leaves the skeleton completely alone. It takes whole mouthfuls of composite material, running them through, extracting the organic fraction, and passing the glass out the other end intact. And the extraction is the part that matters for our purposes, because it's where the containment problem gets solved in a way we haven't seen yet. There's a detailed old study of the alimentary system of a sponge eating Dorid Geruna tomentosa, and what it found was little evidence for extracellular digestion, which is surprising for a predator. And it fits a broader pattern in this group of mollusks, where a great deal of digestion happens inside cells. Material gets taken into amoeboid cells and into the cells lining the digestive tubules, packaged into internal vesicles and broken down in there. Phagosomes, little acid bubbles inside individual cells, doing the chemistry in a volume of a few cubic micrometers. That's the answer. Look at it against the containment problem, and it's almost funny how completely it wins. Hold the enzymes in place? They're inside a membrane-bound compartment inside a cell. There is no leakage, none. Hold the meal in place? The meal is in the same compartment because the cell engulfed it. Recover the product? The product appears in the cytoplasm of the cell that will use it, having crossed one membrane over a distance measured in nanometers. Perfect containment at every stage, with zero loss. The catch is throughput. A phagosome can only handle particles a cell can swallow, which means everything has to be broken down to that scale first, and that's slow, and it caps how fast a large animal can eat. It's also why the big fast predators of the ocean don't work this way. You cannot run a tuna on intracellular digestion, but for a slug crawling across a sponge at a speed you'd need time-lapse to see, throughput has never been the constraint. It has all day. It has all week. Nothing else wants the sponge. So the answer to how do you eat glass turns out to be that you don't. You eat around it, at a scale small enough that the glass is irrelevant, and you let the skeleton go straight through you. And there's a nice downstream consequence. Those spicules leave the slug and settle into the sediment, still intact, still silica. And researchers have pointed out that sponge-eating nudibranchs are effectively moving silicon around the system, taking it out of standing sponge skeletons and putting it into the seabed faster than it would have gone otherwise. A sea slug as a geochemical conveyor belt. While we're on this group, one more thing they do that belongs in a video about containment, because it's the same skill in reverse. Some nudibranchs, the aeolids, the shaggy-looking ones with rows of finger-like projections all over the back, eat cnidarians, hydroids, anemones, sometimes siphonophores, which means they eat nematocysts, loaded ones. Those pressurized syringes we talked about, primed and ready to fire. And the aeolid does not digest them. It moves them, intact and unfired, through its gut, out along branches of the digestive system that extend up into those projections on its back, and stores them there, in sacs at the tips, pointing outward. Then it uses them. Stolen weapons, taken from another animal, transported live through a digestive tract without triggering, and remounted on the thief's own skin. How the slug avoids setting them off in transit is still not completely worked out. And it's one of those problems that gets harder the more you think about it, because these things fire on mechanical and chemical cues, and a gut is full of both. An animal that can carry a loaded syringe through its own stomach without discharging it has a level of control over its internal chemistry that I find slightly unreasonable, which is a good place to turn to the animal that took the syringe idea and built it deliberately. Every octopus den has a garbage pile outside it. That's how divers find them. You're swimming along a rocky bottom, and you notice a scatter of empty shells in front of a hole. Crab carapaces, clam valves, all of it heaped in a way that looks arranged, because it is. The octopus brings food home and throws the packaging out the front door. Biologists call it a midden, which is the same word archaeologists use for the trash heaps of ancient villages. And I've always liked that the term crossed over. Go through one, and the clamshells tell you what happened inside. Some of them are just open. Both valves still hinge together, gaping, undamaged. Some are chipped along the edge, a notch broken out near the margin, and some have a single small hole in them, neat, tapered, a couple of millimeters across at the surface, drilled clean through. 3 different holes in the same trash pile. 3 different methods, and the octopus chose between them. Start with the equipment, because an octopus has more tools for this than anything else in the ocean. There's a beak, hard, curved, made of chitin, shaped like a parrot's, and mounted at the center of the arms, where all 8 of them meet. It bites and it chips. Behind the beak is a radula, the toothed ribbon, the same basic structure the drilling snails use, Behind that is a small muscular organ called the salivary papilla, which sits just below the radula, and the front face of it is covered in very small teeth. This is the drill. And feeding into that whole apparatus are the salivary glands, and the posterior pair of those produce a serious cocktail: enzymes for digestion, compounds that act on the nervous and muscular systems of prey. Octopus saliva is functionally venom, and one of the active substances identified in it is tyramine. Then there's the rest of the animal. 8 arms, lined with suckers, each sucker independently controlled, capable of both grip and fine manipulation, and a web of skin between the bases of the arms. Now watch the sequence, because the order of operations is the interesting part. The first move on a bivalve is almost never drilling. The first move is to pull. The octopus gets the clam into the web, up near the mouth, and applies suckers to both valves and pulls them apart. Straight contest of force. And unlike the sea star, the octopus has real muscle behind it. Work on Octopus rubescens found that how the animal handles a bivalve depends on the shell thickness and on how strong the adductor muscle is, which is exactly what you'd expect from an animal running a cost-benefit calculation on each individual prey item. Thin shell, weak muscle. The octopus wins by pulling, and the clam ends up in the midden intact and gaping. If pulling doesn't work, try the beak on the shell margin. Chip a notch, get an opening at the edge, and put saliva through it. That's the notched shells in the pile, and only when neither of those works does the octopus drill, which tells you something before we even get into the mechanism. Drilling is a last resort. In one study of Octopus mimus in northern Chile, across 70 animals and 9 kinds of molluscan prey, only about a quarter of the prey that got eaten had been drilled at all. So this famous behavior is a minority tactic, reserved for the hard cases. Here's why. Drilling through a shell takes a long time, and every minute of it is a minute the octopus is out of its den, exposed, working on a single meal. When the shell is thin enough to pull, pulling takes seconds. There's no reason to drill unless the alternatives have already failed. When the octopus does commit, the method is the same alternation the snails invented, arrived at independently. The salivary papilla goes against the shell and secretes. The secretion chemically weakens the calcium carbonate at that spot, softening a thin layer. Then the toothed face of the papilla and the radula rasp the softened material away. Then more secretion. Then more rasping. Chemical, mechanical, chemical, mechanical, for as long as it takes. And the result is a hole with a very particular shape. In the Octopus mimus work, the holes were typically a truncated cone with an oval cross-section, wider at the outside and narrowing as it goes in. Measurements of shells drilled by Octopus put the external opening at usually less than 2 millimeters across, and the actual penetration at the inner surface at 1 millimeter or less. 1 millimeter, smaller than the hole in a shirt button. The size scales with the animal and the prey. Bigger octopus, bigger clam, bigger hole, which fell out of the measurements as a real relationship. Thicker shells were also more likely to show holes that never made it through incomplete boreholes where the octopus gave up partway. I like that detail more than the successes. It means the middens contain a record of failure. Shells where an animal spent an hour drilling and then decided the return wasn't worth it, dropped the clam, and moved on. Now hold on to the size of that hole, because the size is the entire point, and it's the thing that makes this the most surgical solution in the video. A millimeter-wide hole is useless for eating through. Nothing is coming out of a clam through an opening that small. You can't get a beak in there. You can't get an arm in there. The octopus has just spent serious effort making a doorway that nothing can fit through, so the hole isn't a doorway. It's a keyhole. And what goes through it is chemistry. The Chilean study did something that most drilling research doesn't bother with. It mapped where on the shell the holes were. Not how many. Where? And for one clam species in particular, Protothaca thaca, the pattern was almost embarrassingly tight. 83% of the holes sat within or right beside a specific band of shell called the myostracum, which is the layer where the adductor muscle attaches to the shell, mostly the anterior one, 83%. On a target that's a small patch on a curved surface, inside a closed shell, invisible from outside. The octopus is drilling at the anchor point of the muscle that holds the clam shut. Once you know that, the rest of the behavior explains itself. The saliva going through that hole lands stressed directly on the muscle system, and on the connection between the muscle and the shell it's pulling against. Compounds that interfere with nerve and muscle function get delivered to the one structure whose failure opens the whole box. And then the clam opens itself. That's the move. The octopus gets the clam to stop holding the shell shut, and after that the shell is just a container with a lid off. So look at what the hole was actually for. That hole was only ever an injection site, positioned with real precision on an anatomical landmark of a completely different animal through an opaque wall. Which raises the question I find hardest to answer, and I don't think anybody has fully answered it. How does the octopus know where the adductor muscle is? It can't see it. The shell is closed and opaque, and in a lot of cases this is happening at night or inside a den. It could be reading the external shape, and shells do carry reliable external cues. The position of the umbo, the growth lines, the overall geometry, all of which correlate with the anatomy inside for a given species. That's the sensible baseline explanation. There's another possibility that got a lot more plausible in the last few years, which is that the octopus is tasting it. Work published in 2020 from Nicholas Bologna's lab at Harvard described a family of receptors in the suckers of octopus arms that detect chemicals by contact. Chemotactile receptors. They respond to molecules that barely dissolve, the kind of compounds that sit on a surface rather than diffusing into the water, which means the arm gets information about what it's touching by touching it. An octopus arm tastes what it holds. Whether that resolution is fine enough to locate a muscle attachment through a shell is not something I'd claim, and as far as I know, it hasn't been tested. What we can say is that the animal has a sensory channel we don't, running through the same surfaces that are gripping the clam, and that the placement of those holes is far too consistent to be luck. Now put the octopus into the framework we've been building because it sits at the top of it. Every animal in this video has been fighting to establish a vessel. The sea star borrowed one. The crown of thorns manufactures a temporary one out of tissue. The larvation builds one out in the water. The worm grows inside one. The cnidarian carries one and sometimes deploys its lining outside. The octopus uses the prey's vessel, unopened, and enters it with a hole about 1 millimeter wide. Think about how little leaks through a 1 millimeter hole. Practically nothing. The shell is still shut, the clam is still sealed, the water outside cannot get in, and whatever the octopus puts in cannot get out. Containment is essentially total, and it costs the animal a hole the size of a pin. The saliva includes digestive enzymes, and there's good reason to think a certain amount of breakdown starts happening inside that closed shell before it ever opens, with the octopus able to draw out tissue that's already partly loosened. How much of the meal is processed that way versus eaten normally after the shell gapes probably varies with the prey and the octopus, and I wouldn't want to put a number on it. The principle stands either way. Deliver the chemistry through a hole small enough that the ocean can't follow it in, which is exactly what the nematocyst does, at a scale about 1,000 times smaller. And it points at something about this entire subject that took me a while to see, so let me put it plainly. Go up on land and external digestion is everywhere. It's ordinary. A spider bites an insect, injects digestive fluid, waits, and drinks the contents out of the husk. Assassin bugs do it. A lot of fly larvae do it, Sea stars are famous for their everted stomachs, but a spider does the same chemistry in a fraction of the time with a fraction of the equipment. Why is it so much easier up there? Because in air, a fluid stays where you put it. Squirt enzyme onto a piece of tissue in air, and surface tension holds it in a droplet. It sits. It has an edge. The concentration inside that droplet stays exactly where it started, because there's nothing for it to mix into. Air doesn't dissolve enzymes. A spider's meal is contained by nothing more sophisticated than the physical properties of water in air. There is no droplet in water. That option does not exist. Release fluid into fluid and it mixes, and the mixing is fast, and the mixing is permanent. That one physical difference is the whole reason marine external digestion looks the way it does. On land, external digestion is a chemistry problem. In the ocean, it's a containment problem first and a chemistry problem second. And every animal we've looked at spends most of its equipment budget on containment. The spider gets a free vessel from surface tension. Nothing in the sea gets that, which is why the last case is such an odd one to end on, because it belongs to animals that have looked at the containment problem, at all of this machinery, and opted out completely. No vessel, no enzymes, no digestion at all. Here's a fact about the ocean that doesn't get said enough. Seawater is not water with things floating in it. Seawater is a solution, and a large fraction of what's dissolved in it is organic. Molecules that used to be alive. Amino acids, sugars, fragments of protein, pieces of cell wall, compounds nobody has fully characterized, leaking out of everything that dies, everything that gets eaten sloppily, everything that excretes. Add all that carbon up across the whole ocean, and the number is startling. Around 660 billion tons of carbon in the form of dissolved organic material. Best estimates put it near 662 petagrams, with an uncertainty of about 30 either side. To put that in context, the carbon sitting in the atmosphere as carbon dioxide right now is in the same range. Same order of magnitude. The dissolved organic carbon in the sea is one of the great carbon reservoirs of the planet, and it holds well over 200 times as much carbon as every living thing in the ocean put together. So the sea is a soup, an enormous planetary soup. And here's the cruel part. It's so dilute that for almost everything alive, it might as well not be there. Take free amino acids, which are the useful fraction, the part an animal could actually build tissue from. In normal seawater, those sit at concentrations in the range of tens of nanomoles per liter. Let me convert that into something you can feel. Tens of nanomoles per liter of a small amino acid works out to a couple of micrograms in a liter of water. As a rough comparison, that's in the neighborhood of dropping 1 sugar cube into 1,000,000 liters of water, half an Olympic pool, give or take. Now try to eat that. There's no particle to catch. A net with a mesh of 200 nanometers, the finest thing in this video, would let every one of these molecules straight through, because they're 1,000 times smaller than the mesh. Filtration is not on the table. Neither is chasing, neither is trapping. Neither is any strategy that involves the word prey. Which is why for most of the 20th century the assumption was that this pool was simply unavailable to animals. It belonged to bacteria. Bacteria are small enough that the physics works for them, and they have transporters. And that was the end of the discussion. Then people started measuring properly. In 1982, a paper appeared in Science from Donald Manahan, Stephen Wright, Grover Stevens, and Michael Rice. They worked on the blue mussel, Mytilus edulis, and they asked whether it takes up dissolved free amino acids from real seawater. Not from a spiked laboratory solution at concentrations 1,000 times too high, which is how a lot of earlier work had been done, and why a lot of earlier work was disputed. From natural seawater, at natural concentrations. The answer was yes, and the details are what make it remarkable. Water passes over a mussel's gill in a few seconds. That's the whole contact time. because the animal is pumping continuously to breathe and to feed. In those few seconds, measurable amounts of specific amino acids were removed from the water, and the removal kept working all the way down. It was still going at ambient concentrations as low as 38 nanomoles per liter, which is down at the bottom of the range you find in open coastal water. So the muscle is pulling molecules out of a solution that dilute during a few seconds of contact using a surface that's already there for other reasons. The machinery is transporter proteins in the gill epithelium. They grab the molecule and haul it across the membrane, and they do it against the gradient, which deserves a moment because the gradient is absurd. Inside the muscle's cells, free amino acids are abundant. Marine invertebrates use them as internal osmotic filler, keeping their cells in balance with the salt outside so intracellular concentrations run in the millimolar range. Outside, in the water, nanomolar, millimolar inside, nanomolar outside. That's a difference on the order of a millionfold, and possibly more, and the transporter is moving molecules from the dilute side to the concentrated side. Uphill the whole way. It manages it by coupling the ride to sodium. Sodium wants to flow into the cell because the cell keeps its internal sodium low by spending energy, and the transporter lets sodium in only if an amino acid comes with it. The energy in the sodium gradient pays for hauling the amino acid uphill, which means the muscle is, in a real sense, eating without eating. There's no mouth involved, no particle, no digestion, no enzymes released anywhere. Molecules of food arrive at a membrane and get pumped inside. Look at that against the containment problem, and something clicks. The containment problem exists because digestion produces small molecules in the wrong place, outside the animal, where they can be lost. If the small molecules are already in the water and you have transporters, then there's nothing to contain. The loss has already happened to somebody else, long ago. You're at the bottom of the process, picking up what everything else spilled. No vessel required, because you're not trying to keep anything in. Pathways for taking up dissolved amino acids directly across body surfaces have now turned up across an enormous range of marine animals. Sponges, cnidarians, flatworms, brachiopods, mollusks, ribbon worms, annelids, echinoderms, arthropods, chordates. Reef corals take up dissolved free amino acids from seawater. Larval echinoderms do it. It appears to be a general property of soft, permeable marine animals rather than a specialty. And then in 2011, a study extended it somewhere nobody expected. Chris Glover and colleagues published work on the Pacific hagfish, Eptotretus stoutii, in the Proceedings of the Royal Society. Hagfish are vertebrates, distant ones, an ancient lineage, but they're on our side of the family tree and they have a proper gut. They're also scavengers, and their scavenging style is specific and unforgettable. A hagfish finds a carcass on the seafloor and burrows into it. Not up to it, into it. It works its way inside the body cavity and feeds from the inside out. Now think about what the water is like in there. Inside a decaying carcass, in an enclosed space, surrounded by tissue that's breaking down. The dissolved organic content of the water goes through the roof. It's rich. It's the most concentrated organic soup you'll find anywhere in the deep sea, and the hagfish is bathing in it. So Glover's team asked whether hagfish skin and gill can take nutrients directly out of that water. They can. Both surfaces absorb the amino acids alanine and glycine, and the pattern of absorption showed the signature of specific transport rather than simple leakage across the membrane, because the uptake saturated as concentration rose instead of climbing forever. It was the first documentation of that kind of direct organic nutrient uptake in a vertebrate. And the authors ran the arithmetic on surface area, which is where it gets properly strange. A hagfish is a long animal with a lot of skin. Their calculations suggested that skin, given its area, could take up nutrients at rates in the same range as the digestive tract and possibly faster. A vertebrate, lying inside a dead whale, absorbing food through its skin at a rate comparable to its own intestine. That's the far end of the ladder. Nothing is caught, nothing is contained, nothing is dissolved by any enzyme the animal made. The food arrives already in molecular form, and the animal's outer surface functions as the absorptive membrane. The ocean did the digestion. The animal just turned up with the transporters. There's a limit on this, and it's worth naming because it explains the distribution. The rate of uptake depends on how fast molecules reach the surface, which depends on concentration and on how quickly the thin layer of water clinging to the animal gets refreshed. At open ocean concentrations, that rate is low, and for most animals, dissolved uptake is a supplement, a useful trickle, not a living. It becomes a living only where the water is locally rich, inside a carcass, down in sediment-poor water, in the fluid around a hydrothermal seep. The soup is everywhere. It's only thick in a few places, and that's precisely where you find the animals that gave up on digestion. So far, we've been following the outbound leg. Get the chemistry to the food, keep it there, keep the food there. Now the return leg, which almost nobody talks about, and which I'd argue is the part that actually decided how all of these animals ended up looking. Because here's the situation after the enzymes have done their work. You've got a thin film of fluid, full of small molecules, amino acids, sugars, fatty acids, peptide fragments. It's sitting inside a shell, or under a stomach, or in a root cavity, or wrapped in a filament, and it's still outside your body. Every one of those molecules has to cross a membrane to become part of the animal. And there are exactly 3 ways to make that happen. Every solution in the ocean is some combination of these 3. Method 1, bulk transport, move the fluid in volume into an enclosed space where absorption can happen at leisure. Method 2, ciliary transport, beat the fluid along a surface with hair-sized motors steering it where you want it. Method 3, membrane transport. Pump the individual molecules across, one at a time, with transporter proteins. Fast, cheap, and selective. And like most engineering menus, you can't have all 3. Start with the sea star, and with a fact about its anatomy that reframes the whole strategy. The everted stomach does absorb some material directly. The bulk of the absorption happens somewhere else entirely, which surprises most people, because the everted stomach is the famous part. The real absorption happens after retraction. When the meal is liquefied, the cardiac stomach contracts and pulls back in, and the neuropeptide we met earlier, NGFF-yamide, is the signal that triggers it. The stomach comes home, carrying the slurry with it. That retraction is a bulk transport step. It's the sea star swallowing what it made. From there, the fluid moves into the pyloric stomach, and from the pyloric stomach into 5 ducts, one running out into each arm, and each duct opens into a pair of long branching organs called pyloric caeca. Cut open a sea star arm, and the caeca are most of what's in there. They fill the arm. The animal is essentially a set of 5 digestive glands with a mouth in the middle and some tube feet underneath. And the lining of those caeca is built for one job. Work published on Asterias Forbes described the epithelium as composed of very tall, very slender cells, flagellated, carrying a dense brush border. Unpack that. Tall and slender means you can pack an enormous number of cells per unit of length. Flagellated means each cell is stirring the fluid next to it. And the brush border is a carpet of microvilli, tiny finger-like extensions of the cell membrane, which multiply the effective surface area of that cell by something like 10 or 20 times. Then the products cross into the coalomic fluid, the body cavity fluid that bathes everything, and get distributed from there. So the architecture is 2 organs doing 2 separate jobs. One is an applicator you can push out into the world, deliberately thin and floppy, so it can enter a gap the width of a sheet of paper. The other is a huge, delicate, high-surface area absorber, kept safely inside where nothing can damage it. That division makes complete sense once you notice the conflict. An absorptive surface wants to be enormous, folded, fragile, and undisturbed. A surface you shove into a clam wants to be tough and thin and expendable. You can't build one tissue that's good at both. So the sea star built two, and connected them with a retraction step. The sea cnidarians solved it differently, and their solution is stranger and, in a way, more elegant. Their digestive cavity is also their circulatory system. That's what the word gastrovascular is telling you. There's no blood, no heart, no separate network of vessels. The same space that holds the meal is the space that distributes it. So when a fish comes apart in an anemone's cavity, the products don't have to be absorbed and then loaded into a transport system. The cavity fluid is the transport system. Cilia lining the cavity keep it moving, circulating that soup through the mesenteries and out into the extensions of the cavity that reach up into every tentacle. In a colonial coral, this gets pushed further. The polyps aren't separate animals. They're connected by a network of gastrovascular canals running through the shared tissue, and material captured by one polyp can move through those canals to polyps that caught nothing. A branching coral colony is a plumbing network with mouths on it. Then the final step is the phagocytosis we talked about. Cells lining the cavity engulf particles directly, finish digesting them internally, and pass the results on. Recovery happens at the level of individual cells everywhere, all over the interior surface, at once. Now Osedax, where the recovery problem is the hardest of the lot. The worm has no gut, so bulk transport is not available. There's no cavity to pull anything into. Whatever comes off the bone has to be taken up directly by the root surface, which explains the root architecture. Roots branch. That's what roots are for, in plants and in this worm. And the reason is identical. Branching is how you generate an enormous surface area inside a solid medium. The worm sends roots into bone to make contact area, because every square micrometer of root surface is an absorptive surface pressed directly against the material being dissolved. Distance between where the molecule is released and where it's absorbed. Essentially zero, everywhere, simultaneously, throughout the interior of a whale vertebra. And the transcriptome work on Osedax japonicus found the root cells expressing machinery for endocytosis, which means the cells are physically drinking the surrounding fluid, taking it in as little membrane-bound packets, rather than relying entirely on individual transporters. Then the symbiotic bacteria in the root tissue do their share of the metabolic work, and the worm takes nutrition from them. Every one of those steps happens inside the bone. There is no point in the entire process where anything valuable is in open seawater. Then the far end of the menu, the mussel and the hagfish, where recovery is the only step there is. No bulk transport because there's nothing to swallow. No fluid to steer because the fluid is the entire ocean. Just transporters, sitting in a membrane, pulling individual molecules across against a gradient of 1,000,000 to 1. And that approach has one specific enemy, which is worth naming because it explains why cilia keep showing up in this story. Water next to a surface doesn't move the way the water further out does. It clings. There's a layer right against any surface, sometimes called the unstirred layer, where flow drops off towards zero, and the only way molecules get across is by diffusion. Now remember how slow diffusion was. A protein needs an hour to cross 1.5 millimeters. Small molecules are faster, but the principle holds. So an absorbing surface eats the molecules in the layer right next to it, and then it's sitting in depleted water, waiting for more to arrive by diffusion from further out. And that wait is the rate-limiting step. You can have the best transporters in the ocean and it won't help you because your problem is delivery. The fix is stirring. And at the scale of a cell, the way you stir water is with cilia, which is why they're absolutely everywhere in this subject. Cilia on the pyloric ceca of the sea star. Cilia along the mesenterial filaments of the anemone, moving product back toward the polyp. Cilia on the dorsal groove of Chaetopterus carrying the food ball forward. Cilia hauling in the mucus web of a pelagic snail. Cilia driving the casiosomes of an upside-down jellyfish through open water. That last one aside, they're all doing the same job, refusing to let the water next to a surface sit still. So here's what I take from the recovery step, and I'll flag that this is my reading rather than a quoted conclusion. The recovery method is what decides the shape of the animal. The enzymes are nearly interchangeable. Proteases, lipases, amylases, more or less the same toolkit across the whole ocean, because there are only so many ways to break a peptide bond. The chemistry is not what makes these animals different from each other. What makes them different is the return leg. The sea star has two organs because absorption and application need incompatible tissue. The worm has roots because absorption in a solid needs branching. The coral has canals because absorption in a colony needs distribution. The nudibranch went intracellular because absorption from a composite needs a compartment small enough to sort. The hagfish has skin transporters because absorption from a soup needs area and nothing else. Start from the question of how the product gets home. And every one of these body plans stops looking exotic and starts looking like the only reasonable answer. Let's try running the logic backwards, because that's where this whole thing pays off. Forget the animals for a second. Imagine somebody hands you a description of a piece of biological equipment and asks you to guess what it eats. Here's a thin, loose sheet of tissue that can turn inside out through a small opening, attached to a set of enormous branched glands that fill the limbs. You can say quite a lot about that animal's diet without being told anything else. The thin sheet means it needs to enter narrow spaces, which means the food is inside something rigid with a gap in it. The big internal glands mean absorption happens after retrieval, in bulk, which means the meal arrives as a fluid. So the diet is animals that come in sealed, hard containers, or animals spread as a film across hard surfaces, bivalves and coral. Here's a set of flexible cords packed with gland cells, cilia, and pressurized microscopic syringes. Syringes only work on things you can puncture, so the food is soft-bodied. Cords mean the digestive surface goes to the meal instead of the meal coming to the surface, which suits prey too large or awkward to swallow. And the delivery is by injection, so the chemistry lands inside the prey where nothing can dilute it. Diet: soft animals, up to and including fish, plus anything organic that lands nearby and can't fight back. Here's a disposable structure with a mesh finer than a bacterium and its own plumbing. Nothing that size lives on anything but suspended particles. And a disposable filter means the food supply is continuous, but the machine clogs, which tells you the animal is in open water in a place where particles never stop coming. Here's a tissue with proton pumps mounted on its outward-facing membrane and a habit of growing into branches. Acid means mineral. Branching means the food is distributed through a solid rather than lying on a surface, so the meal is inside a rock, and the animal grows into the rock to reach it. Here's a millimeter-wide drill and a gland producing compounds that act on muscle. That combination only makes sense against armor that's held shut by a muscle. There'd be no reason to build it for anything else. Here's a rasping ribbon and a digestive system that works inside individual cells. Intracellular digestion means the food arrives mixed with material the animal can't process, so it needs to sort at the scale of particles. Something composite. Something with a skeleton running through the edible part, a sponge. And here's a large, permeable skin covered in transporter proteins with no specialized feeding apparatus at all. Food already in solution, which means the animal lives where the solution is thick, and there aren't many places like that. So you're looking at a burrower, a carcass specialist, or something living in sediment. Every time the equipment gives away the diet. The containment problem admits only so many solutions, and each solution fits one kind of meal and fails at the rest. That's the thing worth taking away from all of this. External digestion is a family of answers to a single engineering problem, and the problem is containment. Where do you put the reaction so the ocean doesn't take it from you? Borrow a container. Build one out of tissue and press it against a surface. Weave one out of slime in open water. Grow into one by dissolving your way in. Carry one and push its lining out through the door. Shrink it down to a bubble inside a single cell. Make a hole in someone else's and inject through it. Or give up on containers entirely and drink the ocean. 8 answers. One problem, now a request, and it's a specific one. Somewhere in your house or your parents' house, there's probably a jar or a bowl or a windowsill with beach shells on it. Everybody has one. Nobody ever looks at them properly after the first week. Go find it and check the shells for a hole. You're looking for a small round one, usually somewhere on the flat of the valve, often countersunk like a machined bevel, cleaner than any hole a rock or a wave would make. 2, 3 millimeters. If you find one, you're holding physical evidence of everything in this video. Some snail spent hours alternating chemistry and rasping to get through that wall, and then put a feeding tube through the opening and digested the occupant inside its own shell. You picked it up because it looked pretty. If you find one, tell me in the comments. What kind of shell and which beach? I go through them all, and I'm curious how widely those turn up, because drilled shells are common in some places and almost absent in others, and the pattern is real. If you find one with an unfinished hole, a shallow pit that never went through, that's better still. That's a shell where the predator gave up. And if this was the kind of thing you want more of, the mechanism and the plumbing underneath the highlight reel, hit like. That's the clearest way I have of knowing which of these are landing. Subscribe if you want the next one when it turns up. One last thought to leave you with. We tend to think of a stomach as a place, a room in the body where food goes. That picture works fine for us because we've got one and it stays put. The ocean runs on a looser definition. Down there, a stomach is a condition. A volume of water held still long enough, with the right molecules in it, and a surface nearby ready to absorb the result. Meet those conditions, and you have a stomach. It can be a mussel shell. It can be a film of mucus under a starfish. It can be a tunnel dissolved through a whale's vertebra, or a pocket sealed by a single cell against a piece of bone, or a bubble a few micrometers across inside a sea slug. Fail to meet them, even for a minute, and there's no stomach anywhere. No matter what organs you own, just enzymes going downstream, and a meal you paid for feeding something else. That's the whole game down there. Everything with a gut is holding a volume of water still against a current that never stops trying to take it away. Some animals do it with a shell. Some do it with a membrane. And a few of them do it out in the open, with nothing but a sheet of slime and the patience to wait.